Bioinformatics.
Hi, and welcome to Lecture 3. I'm going to be talking about some of the different sequencing platforms you might run into out there in the real world, as well as how they generate data and what those data kind of look like when you first get them, and how they vary between the two kind of popular approaches. I'd like to start by talking a little bit about the past of sequencing to give you context for what we're doing in the present, and then look forward to where the future is heading. So, really, sequencing starts with this man, Fred Sanger, who won a Nobel Prize, one of his two Nobel Prizes, for developing Sanger sequencing. You've probably used Sanger sequencing in the past if you've ever done PCR in a lab class, amplified a gene and then sent it off to get the sequence back. That was Sanger sequencing. What I think is really interesting and worth noting about Fred Sanger is that he only published 100 papers in his career, yet won 2 Nobel Prizes. So sometimes it pays to put out lots of little tiny things, but just several things of significance. And so it's not about how much you do, it's about the quality of what you do. So most of you are probably familiar with Sanger sequencing already. It starts with polymerase chain reaction. And polymerase chain reaction, as just a quick refresher, you start with your whole DNA, you have primers, and you're interested in sequencing in between those two priming regions by extending between both different primer sets. And ultimately, you end up with many fragments that are all sequence data between those two primers, often around 1,200 base pairs or so worth of information. And so, how do these many fragments or amplicons, amplified fragments from an original template, end up getting sequenced? Well, without getting into too much detail, at every single one of the rounds of your PCR, some of your fragments get terminated early during that extension phase because a subset of the nucleotides that you put in there to be used for extension actually terminate and also have a fluorescent tag attached to them. So here you can see one that terminated at a G, one that terminated at a T, and the hope is that you have many amplicons that represent all possible size stages from one base pair to the full length of your region of interest. And so you take that pool of those different sized fragments or amplicons and you run them in what's really a fancy gel in the form of a really long capillary tube. And just like in a gel, the smallest fragments move the fastest, so you end up with the smallest to largest going through this kind of laser in order. And that laser excites the fluorescent tag at the end of these, and they're in different colors based on the base pair. So if pink here is G, and red is T, we could see here that in order, we had a T, then a G, then a G, then a T, and then a C. And so this is how the sequence of that fragment of interest is reconstructed. So this went through, 2 decades of optimization, where originally one person could sequence on average 700 bases a day. For reference, it would take you over 100,000 years to sequence the human genome at that rate. All the way up to really at the height of PCR and Sanger sequencing as kind of the primary method for genome sequencing. You could get around 400,000 bases a day, but that would still take 205 years for a single person to sequence a human genome. And so something was needed as far as a change. And that's where today's platforms really got their start. Right now, the two big kind of what we think of as next-gen sequencing technologies out there are run by Illumina and PacBio. There's others out there, but those are kind of the big two you see the most. And I'm going to talk about both of them. So we'll start with Illumina because this is far and away probably the most cost-effective as well as the most common sequencing you're going to see out there these days. And I'm going to walk you through the 3 major steps, which are library preparation, clonal amplification, and then the actual sequencing process, how you get the actual base pair information. So library preparation is just how do you prepare your raw DNA or RNA for sequencing. And so if you start with a bunch of raw DNA, you actually have to cut it down into equal sizes. Illumina is limited to kind of shorter reads, anywhere from 100 to 300 base pairs or so. And there's a little more nuance there, but let's say we wanted to cut all of our base pairs down to about 300 base pairs in length. And then we need to repair them to make them smooth again on both ends so that we can very precisely nick off just one base pair off each end so that we have kind of what we call a sticky end to ligate on adapters. And I'll talk more about what adapters are, but just know that adapters contain a host of things. One, complementary sequence to strings of nucleotides within the actual sequencing flow cell, as well as indexing or barcode information that I'll talk about. So you can get all of your libraries prepped, and then you spread them onto a flow cell. And so this is one type of a flow cell. And at the bottom of this flow cell are all of these little strands of nucleotides that are complementary sequences to the adapters you ligated onto your sequence to your DNA. And so what happens is when you spread all that DNA on there, those adapters immediately go and complement and hybridize with the complementary sequence at the bottom of the flow cell. And so the first step is you start doing a bunch of washes across this flow cell. And so you wash away one strand so that you're left with a single strand that now wants to hybridize or pair with something complementary. So it bends over and pairs with the other side of the adapter. And you end up with this bridge. Now this bridge, they undergo an extension phase and add on all these nucleotides to make it double-stranded again. And then you repeat by denaturing the two strands, and they go boing, and they pop straight up. And now you have gone from one copy of that sequence to two copies. And if you repeat this many, many times, you end up with thousands of copies. And this is really, across the flow cell, almost analogous to a plate in micro, where every single one of these dots really started with a single bacteria or fungi. And so they should all be pretty much identical within these colonies. And so these kind of big colonies of sequences are probably all identical as well. And so you end up with these kind of regions for every single different fragment you started with. And so the way you turn this into actual sequenced information, you take a photo of the entire flow cell. Now, all the base pairs on there are actually going to emit a different color. And so you're going to take a photo of just the very top base pairs. So if we look here, we're looking at a bunch of different colonies of those fragments that were amplified. And in this colony, the top one is G. And in this one, the top one is C. Then you do a wash and you cut off the top base pair of all the fragments in the whole flow cell. And you take another picture. So now you're taking a picture of the second one down. And here the second one down is A. And then you do it again, and the third one down is G. So you just kind of slice your way all the way down through the fragments, and you end up with your sequences that way. And so you end up with lots and lots of short fragments that really reflect those different colonies of fragments, starting from the top of the sequence all the way to the bottom. Now, that washing and cutting and washing and taking pictures starts to lose efficiency after a certain number of times. And so that's been optimized over time. It used to be around 50 base pairs was kind of your limit. Now it's up to about 300 or more. And so still relatively short, but you get many, many of these. You get 400 to 800 million of these different fragment sequences when you're done. There's other ways to kind of extend this and work a little smarter to get length information. Say you really didn't want short read information because you were trying to reassemble a genome, and there were, we'll talk more about the difficulty of assembling over repeat regions. But let's say you wanted information on 400 base pair fragments, but you could only sequence 100 base pair. Well, this is where paired end sequencing comes in. You start with a really longer fragment, so 400 base pairs. You put on your adapters, and then you sequence from both ends. Paired sequencing. There's two pairs of sequencing going on. And so you get information from here, and you also get information from here. Now, this started out as a 400 base pair fragment, and you sequenced 100 from each end. So you end up with this information where you have a sequence that's 100 base pairs, and then you have a sequence that is another 100 base pairs. And they're pairs because you know that they are separated on the genome by a fixed distance of 200 base pairs because it started out as 400. And so this can be very informative. The other kind of nice thing that you can do now with these types of sequencing approaches is, I mentioned when you're doing library prep, you can put on adapters in addition to barcodes or indexes. And what barcodes or indexes really are basically just labels that you can put on your different fragments. So I mentioned that you can get 400 to 800 million reads per sequencing run. Let's say you only need 200 to answer your question. Well, why spend all that money and get twice as much data when you could answer it for 2 different species, for example? So let's say you had some DNA from one species and another. Well, during library prep, you could add onto the adapter a unique serial code using nucleotides. So here, CATTCG to one species, and in the other, AACTGA. And so now you can pool them together and sequence them as if they're one thing. And you end up getting your output, but at the front of every sequence is going to be that index or barcode sequence that you attached. And it will be very easy bioinformatically to separate those into 2 separate files. And now you have a file of sequences from one species and a file of sequences from the other species. You could do this with 96 different things if you wanted. It really just matters how much actual sequencing you want done per set of things that you're trying to break apart. Okay, so that's what we would call kind of short read technology. The next generation sequencing is really long read single molecule sequencing, where you're not dicing up the whole genome and sequencing at random. You're trying to read through large single strands of DNA in one go. And so really the most popular of these is PacBio, or Pacific Biosciences. Now, this is basically a chip that has lots of little tiny wells in it. And at the bottom of each well is a polymerase. Now, what this polymerase does is it grabs onto a piece of DNA, hopefully a very long piece of DNA, and it just goes through Pac-Manning its way around, adding on complementary bases. And every time a base is added, it emits an illumination related to the base pair that was added, whether it was an A, T, G, or C. And so here you can see the intensity there of A versus T in different colors. And so that illumination and the order of that, kind of similar to Illumina, how it takes a bunch of different pictures, will be translated into your actual sequence for that fragment in that well. Now, historically, it has a little bit of a higher error rate. 1% to 2% is actually kind of high. But there are nice ways around that. And the trade-off also, though, is that you get really long reads. You can get up to 10,000 base pairs sequenced from one strand, much more than 300. The other trade-off is that you tend to get a lot less sequencing. Rather than 800 gigabases of information, you might get 5, 10, or 20 gigabases of information. But it's really good, long quality information. And so I mentioned that high error rate. Someone came up with a great way of dealing with that because the errors are distributed at random during PacBio sequencing. There's no error bias. Someone put hairpin adapters during library prep so that your double-stranded DNA actually forms a circle. And so what happens is the polymerase will Pac-Man its way all the way around multiple times now. And so if you look at the sequence in a linear fashion, it really just looks like this circle multiple rounds unraveled. So you've got your adapter, you've got your template strand, adapter, coding strand, adapter, template strand, adapter, coding strand, adapter. And you can stack those up bioinformatically and then look at if there's an error right here on one but not on any other spots. You can derive a consensus sequence that you're really confident has very low rates of error. So that's really an advantage of PacBio. And PacBio, unlike Illumina, doesn't have a fixed length. Every single well is going to give you a slightly different length, and there's ways to optimize it. But here you can see kind of a histogram of all the way from the most frequent lengths all the way down to what you'll get a subset of your reads that can be really, really large, up to, some people even get 70 KB every once in a while. So what's great about PacBio is that it's perfect for genome assembly because long reads are essential for genome assembly, as we'll learn later in the semester. It's also relatively affordable as far as sequencing small genomes. You can sequence bacterial genomes like nothing. Human genomes are still a little bit bigger and plant genomes bigger and complex, but If you're looking at smaller genomes, PacBio is perfect because you can almost read through the entire genome in several reads. So the next thing coming out, that's out now but is constantly up and coming, is Nanopore, Oxford Nanopore Technologies. This is a totally different take on how to sequence. We're no longer looking at fluorescent images. What we're doing here is using a hemolysin pore which has a really small diameter, just enough for one strand of DNA to really fit through, and using an electric gradient, just like in a gel, it draws the DNA strands in their entirety through this pore. And there's many of these pores on any given chip. And as it goes through, every base pair emits an electrical current. And the electrical current varies by the base pair that's going through. And so you can read through and get your sequence data that way as well. And that's kind of what it looks like initially. Now, what's cool about this is that you can get really, really long reads. You can get up to 1 million base pairs in a single read or more. And it's really cool because it's not that expensive. The instrument is only about $900, and every run is about $500 last time I checked. Not only that, it's on a USB chip. You can sequence this in your classroom, at your house, and you don't even need strong computational software to do the initial calling of bases, because it's actually sending all the electrical current information to a remote facility where they call it for you and then send you back your sequenced file, just like you would get from Illumina. And so this is, I think, really kind of the future of long read technology, assuming they get their error rate down. It's really portable. It can be used with varying qualities of DNA. It's even been used to sequence on the International Space Station. And when Zika was a big deal, it was used real time on the ground to try and identify different variants of Zika as that was going on. So it was actually very good because there was such a short turnaround in getting the data. So those are kind of the main ways we get next-gen sequencing data. But it's important to think about what you're going to sequence. And so how you prepare your data for sequencing really matters. There are all sorts of different ways to prepare your data for the same type of sequencing that will give you totally different pieces of information. So you could do things like targeted amplicon sequencing. You could do RAD-seq. You could sequence the transcriptome. You could do hybrid enrichment. And I'll kind of talk about those. So many of you, if you took classes here at Sac State as an undergraduate, you probably at some point dealt with 16S amplicon data. Now, what 16S amplicon data is kind of a hybrid approach of the old school Sanger sequencing, where you start with PCR of specific regions. So you would do uniplex PCR is one way of doing it, where you have all your different samples or species and you sequence for 16S, maybe 18S, and something else for all these different species individually. Then you can pool the same samples or individuals if you were doing multiple genes and do library prep on each of them such that each get a different barcode based on the species or sample ID or whatever. And then you pool them together and do some sequencing. And so what you get out are basically sequences that are identified as belonging to one of your original samples for whatever genes you ended up doing PCR on. So the output of it is very similar to Sanger sequencing, but at a much higher throughput. And so that's probably something many of you are familiar with looking at least the output of those data. Transcriptome sequencing is hugely popular because it's not only a good way of seeing what genes are doing in an organism, how we can look at the level of expression, what's on, what's off, what's on more, what's on less, but also because it's an example of reduced representation. Genomes are very large, and if you want to sequence a lot of things, you can't. If you're sequencing pieces at random across genomes, you can very quickly require high amounts of sequencing to get confidence in reproducible areas of the genome. So making sure that you're comparing gene A to gene A between different species. If a lot of your sequence is getting eaten up by non-gene regions, it might be hard to get the information you want. But if you sequence RNA, you're greatly reducing the amount of different things to sequence because coding regions make up a very small percentage of the genome. Other ways of reducing representation so that you can try and sequence many, many things and get comparable units is hybrid enrichment. This is called a variety of things like anchored enrichment, ultra-conserved element enrichment. In a nutshell, what it is you develop probes that are complementary to some highly conserved areas across whatever your group of interest in is, whether it's a family, whether it's a genus, whether it's an order. And you attach really the equivalent of kind of magnetic beads to these probes, and you toss those probes in with DNA of your individuals, and then it binds to regions of interest, and then you pull down just those areas that binding happened. And so you end up with a subset of the genome that is only close to those areas that you probed for. So then you can sequence and multiplex many, many things and still sequence the areas that you want quite deeply and confidently. So all of these different approaches have pros and cons and ideal uses and unideal uses, and it's always important to think about what you're going to sequence, how you're going to sequence it in tandem. So some questions you should always be asking yourself is DNA or RNA better for my question? Is there already a genome available? If there is a genome available, maybe you don't need long reads to reassemble it. You can just use short reads. How closely related is my reference species to my species of interest? What type of samples do you have? Can you get high quality DNA for long reads, or are you limited to short read because your DNA is poor quality? How many samples do you need? What's your sample size? How many genes are you interested in looking at? Do you want to look at everything? All sorts of good questions. Okay, so let's talk about what your actual output data from sequencing kind of looks like. And I'll be using kind of a short read sequencing like Illumina as the examples here. So we're going to talk about what the format you originally get. And so the format you get is FASTQ format. You end up with this really kind of interesting looking format. And I'm going to break it down, but just know that for every single one of these that I'm going to show you, you typically get 400 to 800 million of them in a row in your output file. And so FASTQ format is a 4-line format. Every sequence or fragment that you have is going to consist of 4 lines. There's going to be a sequence ID line, the actual sequence, a separator, which can be just a plus, and then quality scores that are associated with every single one of the bases that they're beneath. Now, some of these are more useful than others. The identifier line, that first line, can have different pieces of information. It's very specific to whatever sequencer you're using. It can have information like what was the instrument's name, what was the run ID, where within the picture was the actual cluster that you're seeing the sequence of. Is it a pair or unpaired? And then what's the index sequence? So if you were barcoding. The sequence itself is self-explanatory. That's the base pairs that were sequenced. And the separator is self-explanatory. It's just a spacing line that can have a plus. It can have other characters on it too, but typically it's just a plus. And then the real funky thing is this quality score line. These quality scores are based on a Phred scale. Now, before I explain what that is, let's think about why it looks so funky. So if we're interested in p-values, our confidence for each one of these bases called, how hard would it be to interpret this if it was multiple digits for every single one of those base pairs? If you had a G here and then a 0.001, it would shift everything, and it would be really hard to make nice, easy comparisons. So a Phred scale is really a fancy way of condensing p-values down into one character. And so what it is it's going to be based on Q, which is kind of already a condension of p-value, where it's minus 10 times log 10 p-value. And so you don't have to memorize all this stuff, but I'm just trying to give you some context for the why. So if we had a p-value of 1, which is really bad, right? Our log 10 p error would be 0, and our Phred quality score would be 0. if we had a p-value of 0.01, our log 10 p error would be -2. Times 10 would be 20. Or sorry, times -10 would be 20. Okay, all the way up to 40 is 0.0001. Typically, we want our scores for quality and sequencing to be above 30, but greater than 20 can be good enough for a lot of uses as well. Anything below 20 is not something we can rely on. And so how those quality scores, those often 2-digit quality scores, get converted into these 1-digit kind of gobbledygook is called, sorry, let me start over on that one. And so there has historically been different formats for how you convert those quality scores into these single digits. Pretty much everyone now has settled on what we call FRED33. So FRED33 is based on the ASCII table, which, if you look up, has a decimal value for every single character on your keyboard, starting at 0 all the way to 127. Your backspace has a decimal value of 8, etc. FRED33 basically means that it's the decimal value minus 33 as the quality score. So here the exclamation mark for FRED33's quality score is 33 minus 33. So this is your 0. If you have an exclamation mark, your quality score is 0. If you have a plus, it is a 10. Okay? If you have an E, it's whatever 69 minus 33 is. 36. Okay? Why is it minus 33? Well, if you look closely, you'll see that the first 33 characters aren't actually characters that are easy to type in a line. Having a line feed button as one of your characters wouldn't make sense. So starting at 33 just makes sense for having good characters to look at. And so now we have a single character way of representing many, many different p-values, all the way up from 1 down to 0.000008. So one of the things you're going to be looking at in today's lab is how do we use those quality scores to start cleaning up our data. You'll be looking at something called FastQC as a nice way of visualizing the quality of sequence data. And so sequence quality can be visualized as box plots across the entire sequence length per base. So here at the first base, the average quality is quite high. Once we get towards the tail end of the sequence, the average quality is still okay, but you've got a lot of things towards the 25th percentile that start to dip into areas that are problematic. So particularly these individual sequences, we'd probably want to start thinking about cutting them off to deal with having good, clean data, and we'll learn how to do that today. Something I'd like you to do when you have time, and again, this isn't a paper we're going to discuss or anything like that, but you don't have to read it in depth, but pull up this paper that I have available in the Canvas and give it a skim. Ask yourself something that maybe you haven't thought about before. What makes genomic data reproducible in papers? Would you be able to reanalyze the original data from this paper? Would you be able to repeat this experiment if you couldn't? Where would you find the original data? And do you think that that's something important that should be listed as required? Okay, so that's all I have. You're going to go start working on the lab, learning how to pull down data and how to clean it up and everything. So I'll be there in lab with you, ready to help.